it comes back to the fact that I always tell my teams and my colleagues, drugs do not develop drugs. People develop drugs. So if the people working on the project are either not the right ones in terms of technical skills or personality skills, then unfortunately I've seen some projects not move forward because people misbehaved or made mistakes. And that's, of course, for an oncologist who's trying to develop drugs for cancer, you feel really bad about it. 1 out of 10 molecules that goes to a phase 1 makes it all the way to registration. And so if you lose some, not because of the molecule, it really hurts. For every 10 molecules that you don't try, you could have one drug that may help people 10, 20 years from now. And those people is us. So
that's right.
Because we're in drug development, especially this type of drug development, you're looking at 10 years forward, right? So we're going to be there in 10 years. So people sometimes think, oh, they're making drugs for others, or we're also making drugs for ourselves.
So when we get older and might need the drug ourselves, yes. Hello and greetings. I'm Ravi Sarathi, and welcome to the International Business Today podcast. My guest today is Dr. Giovanni Abbadessa, Chief Medical Officer at ModeX Therapeutics, based in Weston, Massachusetts. Giovanni, it's a delight and a pleasure to have you on our podcast and to talk about innovative drug development at ModeX. Please tell us a little bit about yourself so that our audience can get to know you, and then we can go deeper into the kind of work you do.
Well, thank you for having me. It's a pleasure to be here. So I'm an oncologist by training. I was born and raised in Naples, Italy. I went to med school there. I didn't know during med school what exactly I wanted to be, possibly a cardiologist. But then I was also volunteering with a nonprofit organization at the same time. And I spent the summer of 1999 in west side of India. Our organization has started several development projects. So I spent time with lepers, with orphans, we were working with the Mother Teresa, some of the missions from Mother Teresa, some from Muhammad Yunus, who had won the Nobel Prize for Peace. And so that experience, plus a brief experience in cardiology, oriented me more towards oncology, where I felt I could have a deeper relation with the patient and also do something, explore the unknown, do research, learn. So I spent years as a clinical investigator in Milan. I was testing experimental drugs with patients. Some of them were then approved and came on the market. Then I wanted to learn basic research, basic science. So I submitted a research project proposal to a lab in Philadelphia. It was accepted. I came over. The idea was to stay here 5-6 months. It was 2005, early 2005. Then I decided I wanted to, it would take longer. I realized it would take longer than 6 months to learn gene therapy and transplant human tumors into mice and then treat them with genes and viruses. And so I stayed longer. I enrolled in a PhD in genetic oncology. I was attending Falls Chase Cancer Center once a week. And that path then led me to move to industry. At the end of 2007, I joined a small biotech and I moved to Boston.
Well, now you worked at Sanofi before that, right?
I worked at Sanofi for 7 years until June 2024. Before the 7 years at Sanofi, I was for 10 years in small biotechs.
And what was the experience like in terms of what you learned, especially about when drug trials fail or when the company changes priorities and they decide to cancel projects. What was your experience learning from that and managing that process?
So I think I learned on that topic, those topics, I learned a little bit different and similar things in the small biotech world and the large pharma world. In a small biotech world, usually small biotechs have funds for a couple of years. And they have to renew the funds all the time, right?
Yes.
They don't have revenues. They only sustain their research through investments.
Yes.
So in a small biotech, sometimes projects don't move forward because you run out of money. Sometimes because you're not able to execute well enough and to use properly the money that you have. I think that's a problem. And that's a shame because maybe a good drug may not be developed because of that. Some other times, maybe in a small biotech, sometimes there's lack of expertise that causes projects to not move forward or to be reprioritized. In a large company, yes, of course, funds play a big role, but a large company has a longer span, a longer horizon, much longer horizon, of course. And so there sometimes is more either reprioritization of the strategy that may change priorities and projects, even when you're not sure if the project is good or not before you find out. Sometimes is you have many projects and you need to decide where you put your funds and efforts. And so you periodically look at your pipeline and you decide which projects have the best chance and which projects you should move people from and funds from. Unfortunately, a number of times, projects don't move forward because of people. And that's, I think, the most disappointing part of the job. But it comes back to the fact that I always tell my teams and my colleagues, drugs do not develop drugs. People develop drugs. So if the people working on the project are either not the right ones in terms of technical skills or personality skills, then unfortunately, I've seen some projects not move forward because people misbehaved or made mistakes. And that's, of course, for an oncologist who's trying to develop drugs for cancer, you feel really bad about it. 1 out of 10 molecules that goes to a phase 1 makes it all the way to registration. And so if you lose some, not because of the molecule, it really hurts. For every 10 molecules that you don't try, you could have one drug that may help people 10, 20 years from now. And those people is us. So
that's right.
Because we're in drug development, especially this type of drug development, you're looking at 10 years forward, right? So we're going to be there in 10 years. So people sometimes think, oh, they're making drugs for others, or we're also making drugs for ourselves.
So when we get older and might need the drug ourselves. Yes. Yeah. Now, one of the companies you talked about was ArQule as an example of small companies and trying to come up with the right drug. And can you talk a little bit about that story before we move on to ModeX?
So I joined ArQule after 2 years in a small biotech. I had left the small biotech because the 2008 financial crisis made it really hard to stay there. I stayed 8 years at ArQule, and in those 8 years, we brought 7 drugs to the clinic. At the beginning, I joined ArQule in a clinical development capacity as an MD, but and so soon I took over the clinical development team. We brought to the clinic a MET inhibitor, an EG5, a BRAF, then an FGFR, then an AKT, then another AKT. The MET inhibitor went all the way to phase 3. And I was running the one phase 3, then part of another phase 3, directly and through my team, and then the other clinical projects. I think ArQule is a really good example. Well, I don't know if not every company is so successful as ArQule has been, but it's a good example of how perseverance and resilience is really key in our world. You need scientific skills, clinical skills, people skills, business skills, finance, and a lot of resilience.
Mm-hmm.
At ArQule, when I joined ArQule, it was a publicly traded company. I think the stock value was $3 a share. And when I joined, they just had started the first phase 1. And if I remember correctly, in 2012, our phase 2 in lung cancer was positive, so the stock value went to $6 a share. I kept all my shares because I believed in the company. I believed we could do much more. But if you just sold, you would have doubled your money, right? We brought more drugs to the clinic, and so that molecule went to phase 3, the last stage before market, basically. But again, in oncology, 1 out of 10 makes it to registration. We started 2 phase 3, 1 in lung cancer, 1 in patients with liver cancer. The lung cancer trial was negative. And so the stock went down, and then the liver cancer trial was negative, and the stock went really down. I think we went down to $1 a share. So we lost everything that we had gained. We had to restructure the company. We had to let people go because this type of biotech doesn't have revenues, right? So the money you can spend on developing drugs and paying salaries depends on what the money investors give you. And if your stock goes from $6 to $1, you have much less money to use. So we had to dramatically shrink, move to another building. And I, from 2009 when I joined to 2014 when this happened, I had been in basically almost all the board meetings. And so the board, they decided to cut dramatically basic research, keep the funds to do clinical development because that's the most valuable assets you have. And when they let go most of the research team, they asked me to take over also being head of research of the labs. So I was heading a small team of 4 people in the labs. We went down from 60 people to 4 as in terms of lab capacity. I kept leading the clinical team, and I was also working on medical affairs, investigator-sponsored studies and studies with academia. I was helping with business development, with investor relations, and that's where I realized I was fortunate. I was accepted at the equivalent of the semester in leadership at the Harvard Business School, and I think that really, really changed my understanding. As scientists and physicians, we don't get a leadership training or a business training, and that was literally like turning a light on in a dark room for me. And we can talk about it. I have a few very good lessons. Some things that they teach in leadership trainings here, they resonated very well for me because they're things that I grew up doing. The business school professors told us, it's very good to share a meal with your team because when you share food and you're all eating from the same plate or the same things, you tend to share more with others. And I'm thinking, well, in Italy, we do all the business deals at dinner. That's how we do it. And well, let me tell you, I'm going to bring pizza at the office. And pizza is even better than individual sandwiches. You literally share the same plates, right? So I started doing that once a week. And I started bringing both my clinical and preclinical team together once a week. And people became a little bit more comfortable. They were all scared. We had gone through a deep restructuring. So everyone was scared of being the next. That gave them reassurance. Problem was that part of the leadership of the company was very stressed, understandably so. And so every time I had the team meeting with my preclinical and clinical team, the CEO would come into the room, wanted to hear what we were talking about, and everybody would fall silent. They were afraid. So I think I never told my CEO, but I started organizing my meetings at my house in the morning at breakfast. I started cooking breakfast, pancakes, and brought the team there. And in that way, they were more relaxed. They felt much less pressure. And they told me, look, we were afraid to tell, but we have a preclinical molecule that we think has a merit. But the CEO and the head of R&D and even our partners decided to trash it. So I said, okay, let me look at it. What is it? Oh, it's a BTK inhibitor. They say it's dirty. And so they don't want to bring it forward. They're worried of toxicity. Fair to be told, having been also in a lab during my PhD program, when we work in labs, we focus on our cells, on our benches, on our animals. It's a very focused and data-oriented way of thinking. And that many times translates in how the data is presented. And so when I started looking into this data in 2014, I found pieces of data here and there, not in a cohesive manner. And those scattered pieces of information is what my CEO, my head of R&D, the partners had looked at. But because they didn't have a preclinical background and a training, they saw lots of graphs and data, and also they saw a drug that was dirty. It hit multiple targets, and they thought, yeah, this is not going to work. However, they did have one study in mouse, and the mice didn't die. So I figured, okay, if the mice didn't die, then maybe this thing is dirty, but not so toxic. Let me understand more. So I spent a month or two maybe looking at it, collecting all this data, putting it together, reorganizing it, asking questions to fill the gaps. And then it's like a puzzle. You have pieces of data, you don't know in which order they go, because they do experiments one by one. Sometimes the experiment that goes here was an experiment that was done after. And so you have to move things around. And finally, it made sense to me. There were some holes. But my CEO and the R&D said, no, don't touch this drug. We trashed it. It's finished. It's shelved. Focus on what comes next. Well, there was not much coming next. There were the existing drugs in the clinic that we were improving our understanding on them. So I told the team to start doing some experiments in secret. We discussed the projects at my house over breakfast, and then they would go to the lab and I would go to the office, and they would do the experiments in secret. And to characterize better, not very expensive experiments, to characterize better the understanding of the drug, the targets, and so on. And we saw that this could make sense, but we needed now to do experiments in animals. So this was a BTK inhibitor. BTK is the Bruton tyrosine kinase. It's part of the B-cell receptor. It's relevant in lymphomas, but especially in chronic lymphatic leukemia, CLL. Mm-hmm. And at that time, 2014-15, one drug and then another drug were being developed, ibrutinib and acalabrutinib. And ibrutinib was the first, and it made history. There's books written about that drug, except it binds in a covalent manner. So it binds and it never detaches. When it does that, the BTK on the cell, as a resistant mechanism, generates a mutation on the 481S residue. Our drug was non-covalent. It bound differently, and it would bound in a different place. So it could bound regardless of that mutation, and it wouldn't cause the mutation. So I said, well, if it is well tolerated in a mouse experiment, and we understand how it works and what other targets it eats, guys, maybe we should do more. So I found that an Italian guy in Ohio, Columbus, had a model of a mouse where he took the mouse, irradiated the mouse, took leukemia cells from patients, transplanted into the mouse. And now I could use the mouse model with the human cells to test drugs. So I called him. I say, Professor Croce, you were a professor in Philadelphia. I was in Philadelphia. We're both Italians. Please help me. I need to test my drug. Oh, nice to meet you, Giovanni. Yeah, no, with pleasure. But, you work for a biotech company. You have money. No, we don't have money. We're strapped for cash. Please. So I took a plane. I took a flight. I went to Columbus. He's a very accomplished, very well-known professor. And he was a super nice guy. And we immediately connected. So we agreed on the study design and to test this molecule. So we made he really helped. And so we made an agreement to send him the drug. He tested, it worked. The leukemia was being wiped out. So at that point, I went to my CEO and said, look, who was also Italian. I said, Paolo, I think this thing has maybe some possibility. We should do something about it. Yeah, but the head of R&D and the partners decided that it doesn't work. Well, they say it's dirty, but it's dirty maybe not in a bad way. And we did experiments in secrets in the lab, and we found out what dirtiness means and what the other targets are, and it shouldn't be a dangerous dirty. Besides, the mice are not dying. We will kill the leukemia, not the mouse. Let's see what I can do. So he convinced our head of R&D to allow us to go and check it. And fate wanted that in the same Columbus, Ohio location where I did the preclinical test was a key clinical investigator who had developed ibrutinib and acalabrutinib and was developing acalabrutinib. So one of the guys with the biggest knowledge in the world about BTK was there, John. So Paolo, I said, Paolo, look, you go. So I don't want to be in the middle. Please, you go. You're the CEO. Talk with him, see what he thinks. I'm not going to come, so there's no bias and so on. So he went. The guy loved the drug. He said, what are you doing? You should bring it forward. So finally, I got the permission to do more experiments with the team. And we did more in vitro, in vivo, in animals. And then in 2016, as more patients were being treated with the BTK inhibitors, ibrutinib, it was emerging that leukemia patients were getting transformations. And a leukemia, chronic lymphatic leukemia is a relatively slower disease. And ibrutinib was controlling it. But some patients were then transforming into an aggressive lymphoma. Possibly because a clone was being selected. You control the leukemia clone, and the lymphoma clone maybe take over. This is called Richter's transformation. So I started testing I wanted to see if our drug would work on Richter's transformation. That would be big. We did an animal model, and it worked. We did in cells, in vitro, and it worked. I said, guys, this drug could be useful not just for leukemia, but for lymphomas. This could be really good. So we put more funds, and we brought it to phase 1. And in the end, phase 1 gave very good results. So Merck later on acquired ArQule.
So before you got to phase 3, Merck brought your company.
Oh, yes. We, as I say, we went $3 when I joined, $6 after a positive phase 2 in lung cancer, $1 after negative phase 3. And then just thanks to this BTK inhibitor, we had a very nice AKT inhibitor too in the clinic and an FGFR. But this one got the company acquired for $20 a share.
Nice. Good.
And then, Merck is still developing.
Great story. Yeah.
So it's resilience.
Yeah, right. Resilience.
It's science. It's clinical. It's business. It's finance. It takes a lot. And if any of these don't work out, you may have a molecule that works and it will never make it through. And so, people, if you're not in this world, you think, why are drugs so expensive? Well, because for every 10 that you put money on, only one
actually
will
works.
make it through. And so to make it sustainable, if a company invests in 10 drugs and doesn't recover enough on that one, it doesn't have the money to invest in 10 more. And so the cost of that one has to finance the other 9. That's why that one is so expensive.
Right.
And then cancer in particular is incredibly smart. As the story with the BTK, right? You hit it on one part and the cancer evolves and modifies itself, and then it's able to resist. It's harder to develop a drug for cancer than for a virus.
So good. Let's talk about ModeX, because at ModeX, you're trying to very hard to treat solid tumors, among other things, right?
Solid tumors,
non-small cell lung cancer. Yeah. So, and you're using antibodies, but you're using this technique called multispecific antibodies. Yes. So tell us and the audience a little bit more about how you're working with this specific approach, and why are you choosing this different approach to targeting, solid tumors, other advanced cancers, immune disorders? Let's hear your take on that.
Yeah, and to be clear, I'm not here representing the company.
Right.
Usually, this is a personal conversation on my behalf.
Right.
Scientific, business, finance, everything.
Yes.
So ModeX, we can talk about it, but when I decided I would leave Sanofi last year, I really took some time to choose where to go. I consulted for a couple of public entities to help them improve their healthcare systems. I consulted for a dozen companies, for a couple of venture capital funds, and I wanted to find a company which has more than one molecule, a potential for a pipeline,
Yes.
with smart, good people and good funds. So ModeX was founded in 2020 by Elias Zeruni, who was past director of NIH and then head of R&D at Sanofi, and by Gary Nabel, who was also very senior at NIH until he became chief scientific officer at Sanofi. And so Gary at Sanofi had developed this breakthrough lab, a lab with a group of scientists to make very innovative molecules. And they started focusing on trispecific T-cell engagers. Gary is on one of the original patents for CD28, which is one of the ways to engage T-cells. And so at Sanofi already, they developed molecules that activate T-cells binding on CD3, which is a protein expressed.
That's the signal 1 pathway.
Signal 1 pathway, very nice.
Yeah.
And then CD28, signal 2.
As a co-stimulator.
Co-stimulator, which helps with increasing survival and sustainability of those T cells.
Right. It makes the T cells, it doesn't exhaust them.
Very, very, very good.
Right.
Yes.
Okay. Yeah.
It's one of the mechanisms to avoid exhaustion. The other one could be 41BB. So there's plenty of publications showing that you activate T cells through CD3 binding, and there's drugs on the market already. Recently, especially new drugs have been put on the market. They leverage CD3 to activate T cells. It works nicely to activate T cells. When you do that, however, the T cell goes in a burst of activation and then rapidly goes into exhaustion and apoptosis. It dies. When you also give CD28, you trigger self-survival mechanism. And so the T cell activates mechanism so it doesn't die anymore. Now, the tricky part is that if you give a lot of CD3 stimulation, you activate the cell so much that it starts producing cytokines, which are needed to recruit more T cells to attack the tumor. But an excess in cytokines, especially interleukin-6, causes important side effects. Also, if you stimulate CD28 too much, you have a massive immune response. There was a drama in 2006 in England. A company named TeGenero was testing a CD28 superagonist. They wanted to superactivate T cells through CD28. They put this molecule in 6 healthy volunteers. And at that time, the rules were different. They dosed each one in the same room, I don't know how many minutes from each other. Each one sequentially started feeling sick, difficulty breathing, hypotension. They were having what we call a cytokine release syndrome or a cytokine release storm. They went into intensive care unit. Some stayed there months with sequelae. This is healthy volunteers. They were thinking they were testing and very low dose of a molecule that shouldn't do anything. How did it happen? Because before we bring a drug to the clinic, even in healthy volunteers, we test it in vitro, in cells. Then we test it in one or two usually animal models. Problem is they tested the molecule in monkeys and they were doing fine. Evidently, the binding of the receptor in monkeys is different than in humans, and in humans it had many more toxic effects. So when you use CD28, you want to be very careful. So what Gary's team has done already at Sanofi and now at ModeX is to only use a monovalent, a single tiny stimulation of CD28, no superagonist effect. And we test all of our drugs for that. So now because you have 2 signals, not one, you can afford dampening down both. So through this approach, you can have a very low touch on CD3 to avoid those cytokine release, unlike most of other drugs, and a very, very low touch on CD28, to the point where they made the same version of our antibody. In one case, they removed CD28. In one case, they removed CD3. Our same antibody with only CD3 activates T cells, but they go into exhaustion.
Cell death, yeah.
Our same antibody without CD3, only with CD28, doesn't activate anything, does nothing. When they go together, cells are activated more and survive longer, and they kill tumor cells more. This phenomenon is known in literature. It was published, in fact, by Gary's team on Nature years ago. So at Sanofi, they made trispecific, 1 tumor target and 2 on the T cell. Sanofi then decided they were no longer interested in this technology. Gary decided to leave, Elias left, and they started ModeX in 2020. I stayed at Sanofi until 2024, and I actually brought to the clinic one, and then I was overseeing the other drug as well, T-cell engager trispecific with, and the results were presented already at conferences, were published. It was manageable. Tolerability was manageable. These drugs were not brought forward for different reasons. So when I came in ModeX, I already kind of knew this technology. I knew the people. A bunch of scientists left Sanofi and went to create ModeX, right? And the technology is fully now owned by ModeX. It owns nothing to anyone. So at ModeX, they perfected this and they turned it into a tetraspecific. In fact, in the lab, they were able to make hexaspecific, an antibody that binds 6 targets. In the clinic, we have 4 specificity. The difference between these multispecifics and other multispecifics that others are doing is that most other groups are achieving binding multiple targets by fusing together 2 antibodies or by fusing together the binding site of the antibody, the one that binds to the target,
the ligands,
and they link them together or they fuse them and so on. That has a number of limitations. One, stability. You have to. It's unclear how stable it will be as you go into the bloodstream and then the tumor microenvironment and so on. Pharmacokinetics, the amount of drugs in the blood, the manufacturing, the cost of manufacturing, how tricky it is to manufacture at a large scale. But then also, you cannot play with those binders. The binders are the pieces of the antibody that bind to the targets, right? Whatever the binder is, that is what you're going to put there. In our case, they patented technology to use a normal human antibody of the same weight as a human antibody, not a big thing. And you know how the antibody is like a Y shape, right? There's a long portion called the FC portion and 2 short arms. The short arms are the ones that bind to targets. Normally, a normal antibody has both arms binding to the same target, okay? They patented a way to put 4 binders. Now, not only they patented a way to put 4 binders, but also to move them around. And when you move them around, the way they bind to targets changes. Because if you bind it to, let's say, CD3 and CD28 on the same arm, it binds differently than if CD3 is here and CD28 is here. The affinity, meaning how sticky the binding is, changes. So now, while it takes months to identify a new binder, and you cannot infringe on patents of other people, so it takes months and months and months to find a good binder that doesn't infringe a patent. So let's say you want to do CD3, but you want to detune it. It's going to take a long time to find a CD3 that is low affinity. However, with this method, the same CD3, you put it here, it binds, for example, much less than if you put it here. And so by moving it around and playing with the relative binding, right? It's not just each individual binder affinity, it's how that affinity relates to the other ones. For example, we're now bringing to the clinic, anytime now at this point, a T-cell engager for lymphomas. It binds CD19 and CD20 on the lymphoma cell. These are proteins that are on lymphoma cells and on B cells.
On the surface.
And CD3 on CD20 on the T cell, right? We want this drug to hit first the lymphoma cell and then activate the T cell. You don't want to activate the T cells everywhere and then bind the lymphoma cells. Otherwise, if you activate T cells everywhere, you're going to have cytokines and you're not going to kill. So by moving these things around, now this drug binds much better to the lymphoma cell than the T cell. And this is true for the other molecule that we have too. So this technology is really elegant because it's simple. It's a normal human antibody. You can play with what you put on the FC portion. On the FC portion, you can even fuse other proteins if you want. You can play with the FC portion to extend or shorten the half-life. We're making tetravalent multispecific molecules for viruses, antibodies, and they played with the FC portion to extend the half-life, so you can give it less frequently. For cancer, it's a much shorter half-life. You want to have a short half-life so you can control the toxicity. If there's a toxic effect, as soon as the drug disappears, you improve, your the toxic effect lasts less. So there's a lot you can do with this technology. For cancer, the point is you want to bind more than one target, because just like in the BTK story, when you hit one target, cancer more easily evolves and resists. It's a natural selection. Cancer is not monoclonal, it's multiclonal. So in a let's say a sphere of tumor, you're going to have different type of cells. When you kill one, well, the other ones just keep growing. And so the cancer evolves, and now it doesn't express that target, it expresses other targets. So if you can hit at least two targets, you put more pressure on the cancer, and the response should last longer. In fact, most anti-cancer therapy are combination therapies today, right? So this technology allows you to bind two cancer targets and activate the T cell in a more physiological way, not just with a lot of CD3 binding, but mitigate your CD3 with the CD28, increase the survival. It has multiple advantages, and but it's a new technology, of course. So we need to learn how to use it. We're testing it now in patients. It's public information on clinicaltrials.gov. There's a website where everybody can see any clinical trial. Right. And our first clinical trial with a MET TROP2 CD328 started in the clinic fall last year. Sorry, at this point, 2023. Today, 2024. Fall 2024. Today is 2026. So the fall of 2024, the first clinical trial started.
Now, how many patients do you typically need to recruit once you start working with humans? And what is the timeline? How many years between, healthy people and then sick people and then doing controls and placebos or whatever you have to do ethically to be able to get to the point where you can prove and maybe get approval from the FDA?
The testing of molecules in healthy volunteers is something you do more in other therapeutic areas. In oncology, because of the situation where the patients are, and because of the mechanism usually of these drugs that can be toxic, you don't go in healthy volunteers. You go from animals straight into patients. It's called a phase 1.
Okay.
A phase 1 in a neurology study will be healthy volunteers. You test one dose, a microdose of your molecule, and you check the pharmacokinetic, how long the drug is in the blood, how long it takes to go away, if there's any biological effect at subtherapeutic doses. In cancer, you don't want to give a cancer patient such a subtherapeutic dose. So you do animal tests in 2 different animal species so that you can try to understand what the possible adverse events will be and how tolerable the drug is. And then there are rules and methods to choose a much lower dose. That's what you start in patients with. And you test, and there's different ways of doing what we call a dose escalation. You start at a small dose and you go up. There's different statistical ways to get there. But let's say a phase 1 dose escalation study in cancer patients. Well, it depends on the type of drug, how many patients, because for some drugs, you can start at a higher dose. For some other drugs, you have to start at a lower dose. With T-cell engagers, you start at a lower dose because you can't risk
you don't want that cytokine storm that you were talking about.
Exactly.
Yeah.
Among other things. Yeah. So it could, usually could be 5 dose levels. It could be 9 dose levels. If you assume that each dose level, it takes about 3 months, if you're efficient.
Yes.
If you're efficient, it takes 3 months for each dose level. Then 18 months for a dose escalation study, usually in oncology, but it could be 2 years. And could it be 40 patients? Could it be 60? Once you think you may have a dose that is active and not too much toxic or not toxic, then you want to expand the number of patients you test that dose in. In the latest years, FDA has pushed a lot also to optimize the dose, not just test one dose. And that's because with the older drugs, chemos especially, you had to push the dose as high as you could because a chemo just kills, right? And so you kill as much as you can, and you try to give the maximum dose possible up to a point the patient will have too much toxicity, hair loss is the minimum. Today, with biologic molecules and more precise molecule targeted therapies, that's not always true. However, it is difficult, and I wouldn't say, there's been a dramatic push to try to use the lowest dose. I think now we're rebalancing a little bit, because the truth is the amount of molecule that goes in the blood could be very little relevant to the amount of drug that goes into the tumor. Tumor creates its own vasculature, and it's all disrupted. And so while in your neck, your blood should flow very nicely if you don't eat too many burgers and chips. But in a tumor, it will go very slow and it will stop and very filtrated. And so there's much more drug in the blood than in the tumors many times. So optimizing the dose based on drug values, based on side effects, it takes a number of patients to know if a dose is better than the other one, if both are active. It takes quite a bit of patience. It's not a straightforward thing. But of course, we're all making that effort, because why would you give a patient more toxicity than is needed?
Yes.
It's needed to have efficacy. It's just that to find that balance, it could take hundreds of patients.
So let's talk a little bit about the U.S. environment for developing drugs. We've had some major changes in terms of funding of drug development, in terms of funding. My understanding is California, New York, Massachusetts are three of the biggest sources of innovative academic drug development and drug conceptual development. And so the U.S. government is changing priorities as well as changing, who they fund. So what is happening, in your opinion, to the ability of the country to develop drugs the way they used to be able to?
That's a very wide question. So yes, these states are the ones where the academic groups have received the highest per state amount of funds from NIH, which means they have the highest number of top-level academic centers. Now, when the government gives money to NIH, 80% of the money is actually redistributed to academic centers throughout the country. NIH only keeps, I think, about 20% for its own research. The rest, it functions as a distributor. And so when the government decides to cut the funds to NIH, effectively cuts the funds to academic centers throughout the country. What that means, there's always something good and it's never black and white, right?
Yes.
The mechanism, how these funds have been allocated, because the funds have grown more and more and more over time, and good academic centers have been established, there's improvements in the mechanism of distributing those funds that definitely can be obtained, right? When you just cut the funds, what happens is Boston University, Northeastern, Harvard, MIT, Kansas University, University of Miami, any academic centers in the US gets less money. Any academic center that does research. When that happens, it means academic centers can no longer pay the things that they need to pay to do research. And I know from talking with several colleagues in academic centers that a number of academic centers in the US have cut dramatically the number of PhD students, for example.
Yes.
Right? Some even by half. Now, if you have to cut the number of PhD students by half for the next 3 years, it means that in 3 years, we're going to have half the postdocs.
Yes.
And the postdocs are the ones who do the research. They're the workforce of academic centers. If in 4 or 5 years from now, academic centers want to keep doing research, they will have to import postdocs from other countries. So we're basically cutting funds up there means cutting workforce preparation here and need to import specialized workforce unless we decide we give up on science and research and we let other countries do it for us. It's like deciding to disrupt all a pipeline of workers for highly skilled technology development and something like that, right? So, but not only that, biotech companies, so I think 68% of drugs that go to the market and that are commercialized by large pharma actually come from small biotechs.
Mm-hmm.
And that percentage has increased over the last years. So if small biotechs don't develop drugs, large pharma don't have drugs to bring in, and people will have less drugs for them to be treated, right? Many of these drugs some of these drugs come from startups that come out of academia. So if you're cutting PhD and research and postdocs, you're going to have less startups, less developments, less discoveries, less drugs. We perfect our research and do make our multi-specific antibodies and drugs based on targets and mechanisms that have been discovered and described by academic colleagues. Biotech companies have funds, as we discussed, for a span of 2 years or so. We cannot afford to do research that will only give fruits in 10 years. That's for academia to do. If academia doesn't have the funds to do it and the people to do it, that means that in a few years, we're not going to have the new targets, the new discoveries that biotech companies will need to make drugs on. Not only, even for current targets, academia keep refining the understanding of those targets, just like my team and I refine the understanding of the BTK inhibitor. If academia doesn't do that, I will make less good drugs than I can today. So those cuts the cuts, again, the mechanism should be improved, it can be improved. If we just stop the flow, it means there are going to be less workers in academia, less science, less startups, less drugs for biotech companies can develop, less drugs that large pharma can acquire, less drugs for people. Now, where does it go? It's today, nothing is in isolation, right? So that also means if we just stay in the US and before we look at the rest of the world, that means that if Boston University, Taft, Northeastern, Harvard, they cut by 50% the PhD students. A town like Boston, a city like Boston will have 50% less junior people, workers around. It's those people pay rent, they buy t-shirts, they go to restaurants, they go to the Red Sox games. It's a large amount of people who are not here anymore, they're not employed, and they don't spend. But also, all these universities, what people don't realize, when NIH gives money to, let's say, whatever, X university, right? That university has to use, those are grants, they have to be used to do research. Yes. So you pay PhD students, you pay people, you buy microscopes, you buy reagents, you pay taxes on those. You pay, the people who get the salary, they pay taxes on their salary. They pay rental, they pay taxes on their, people pay tax, they buy t-shirts, those go back. So a lot of this money actually already goes back in taxes because either the academia uses them to buy things, pay electricity, rental, facility, cleaning crews, and so on. They go back in taxes. And then the salaries, they're spent in the city. So a lot of this actually goes back in, only a certain amount of this money is spent, vanishes, right? Then if you don't discover new drugs, if academia doesn't start up new companies, there's less investments locally. For every 10 drugs, what will make it? For every maybe 10 inventions, maybe one can make a startup company. Investors will put private money there. Larger companies will buy the smaller company and generate a financial cycle in the city, which will then bring more money back to develop things. So once you stop it at the source, there's a whole economic
multiplier
consequences.
Effect down the road. Yeah.
Now, in Boston, we have a number of academic centers and the hospitals are connected and so on. So they suffer. But people who live in Boston, they have where to go to be taken care of. If you live in a city or in a state where the academic center is the main provider of jobs in that city, and this is not East Coast states we're talking about, if in that state the healthcare is related to the academic center, if that academic center goes down, the hospital, the healthcare quality goes down. So people in those states, which have less scientists than the Northeast has, they're actually the ones who are going to suffer the most. It's counterintuitive, but we have alternatives to go get an oncologist in Boston. In other states, there's one academic centers with one hospital.
So
that's just looking at the U.S.
Right. So let's talk about China, because you mentioned China as being, first of all, China and the U.S. are in a major global competitive race. So particularly in biotech, you've talked to me about how interesting China is as a country and its national policies for biotech. So let's talk about that. What is your perspective from where you sit as, with more than 20 years of experience in biotech? Where do you sit, and what is your perspective on how China is approaching developing a biotech ecosystem, just as the U.S. is withdrawing to some extent from that long-term effort?
I think they're doing great, and they're doing all the things that we should be doing. Yes, it's a race. I don't know if it's clear to the public and to administrators how important that race is. And in this case, it's not a race to win something, because the product is not cars or when in the 60s they were racing to go to the space.
Yes.
Part of it was to, show that you were stronger, but the practical implications were less maybe direct. In this case, it's a race that makes drugs. Assume that in the 60s, when the US was racing with Russia, against Russia, to go to the moon, they decided to totally defund the space program. We would have not gone to the moon, and the Russians would have, right? So in this moment, we have a race which is to make drugs, which, by the way, it's a large slice of the economy. It generates so much economy around it, but it ultimately makes drugs.
Cures people. It makes lives better.
Treats people, yes. So again, the mechanism can be improved and should be improved. I don't know if the public understands how dramatic the consequences can be if we just give up on it. China has been very smart, and they have been, in the last 10 to 5 years especially, the regional governments have been investing not only in academic center, but also directly the public government has been investing in private companies. Now you would say, but that's a communist country. Well, for the last decades, the government has invested in academia here. We're not a communist country. The government has invested in the firemen and the police and the postal service. We're not a communist country. There are certain aspects of life where the public, our taxes, are used so that we can live better. That's the police. That's the firemen. That's the ambulances. That's the hospitals. That's making drugs to survive and to treat diseases. China has been investing a lot lately. Not only investing in terms of money, but also in thinking. When I started this job, I was an investigator, again, a clinical investigator in 2001, and testing new drugs. We wouldn't think of China at all. No, they were copying molecules. In fact, even less in 2001, they were barely copying molecules. In the 2010s, more. They were copying molecules, drugs, and making them for their own market. So you would approve a drug here, you should do all the work and all the investments here, then they would just copy it and make it. But in the last 5 years especially, if not 10, they started making their own discoveries, their own drugs, because the governments have invested in academia and even in private companies. So we're at the point now where there's metrics out there. The number of clinical trials being run in China is basically the same, if no more, than the ones in the US at the moment. The number of new molecular entities, new drugs, not copycats, new, it has increased. Probably it's at this point higher number than European biotechs and academia make. There's metrics about the number of business deals that global pharma companies have made in the last years. if you look at those numbers, you'll see that while 10 years ago, large pharma would come to Boston, New Jersey, California to look for small biotechs to buy drugs from, the small biotech would take the risk, develop the first drug, make the innovation, and then the large pharma would come and buy the drug to then do larger studies and put it on the market. Now, those large pharma companies are buying a lot of new molecules in China. The number of the amount of money that went from large global pharma companies to China deals has increased each year since 2021, I believe. And at the end of 2025, I believe that the amount of money spent was even a third higher than the amount of money spent in 2024, which was higher than in 2023, which was higher than every year before. So what does this bring? It brings that those are billions of dollars and rights to new drugs. Instead of coming to American startups and new companies and workers, the large pharma companies are giving the drug to the best ones, and the best ones are coming more and more from China. But also it means that, when I was testing drugs in 2001 with patients, usually drugs would be first tested in the US and then they would come to Europe and then in the rest of the world, which means that a cancer patient at a given time would have access to more experimental and new drugs here than in Europe, than in the rest of the world. If the drugs are first made elsewhere, people in other parts of the world will have access to drugs, which, in the end, because we're not talking about putting a flag on the moon, but about making drugs for people, it's okay. Wherever the drug is made, is great, as long as it then makes it to the rest of the market and the patients. But the question is, do we really want to give up on being the top innovators in the world for drug development? Do we really, did the administration in the 60s even consider cutting funds to the space program when they were racing? I don't know that the public understands and sees what happens and the consequences on the economy, on jobs. And you don't want to be a scientist, great, but it doesn't matter. The scientists employed in a number of cities pay rentals. They go to restaurants. If you own a restaurant, you want the scientists to have a good job so that they can come to your restaurant. If you sell potatoes, well, you want people to be able to buy French fries, right? So it impacts beyond the science world. And if you get a cancer, you want to have
access to the most recent drugs available.
To the most advanced drugs and technologies.
Okay, so let's wrap up. Where do you see this going? Will there be a pendulum shift back to saying we really need to stimulate basic science in drug development in academia once again in the US? Or will we, like in solar panels and like in robotics now, will China take the lead and start exporting fairly high-priced, very useful drugs, and we will become even more, indebted in that sense? Where do you see this going?
Well, first of all, one important point, I see Chinese biotechs and health science experts as potential collaborators.
Collaborators, right.
We need to develop drugs. So if I can do it here, I can do it in Europe, I can do it in Australia, I can do it in China. We'll do it where we need to do it. And if they're good, great, let's collaborate. Where this will bring... I think it's a fast race. Okay, I don't know if you follow Formula One, but if you lose a few seconds, you're lost. Because while you stop or slow down, the other doesn't. China has accelerated while US has been slowing down now.
So the gap is perhaps either diminishing between us.
I think they're on the path to take over. I don't think there's many doubts. it doesn't mean that we cannot make it back. In fact, this country has such background and potential and expertise and minds that have been trained for decades that if it wants to put itself back onto it, it can. But if some decisions are not made, and I don't not necessarily to go back to what it was until a year ago. Again, mechanisms can always be improved. The question is, how do we? I mean, you need the will to do it, right? If there's a will from the public at the administration to sit down and think, okay, how do we make it better than it was 5 years ago? And how can we make it better than it is now? Then it can absolutely be done and brought back to be the biggest science economy in the world. But it needs an active will.
Mm-hmm, mm-hmm. So a more educated public and a more educated administration that is aware of the long-term nationwide benefits from investing in basic science and going back to the beautiful ecosystem we've had for 50 years.
Yeah, look at the example of Israel in the 80s.
Yeah,
Israel decided to heavily invest in technology and become a startup nation focused on technology. And they did it. And a lot of innovation and inventions came out of there. They had to do it for self-defense, almost, and to stimulate the economy, which I remember the 80s was not very good. But even a small country like Israel can do it. So imagine what the US can do it if you really put yourself to it. It can be done.
Yeah.
We've been doing that. This country has been doing it for decades. It just requires will and understanding. And more educated public, more educated administration. I think part of the problems today is communications and the way that people communicate. If you have knowledge, well, sometimes we all speak without realizing that other people may interpret what we say in a different way that we intended. And I think there's a problem with scientists and the science community. The more educated in certain aspects you are, the more you give for granted what you're saying because you know about it.
Mm-hmm.
But you should always remember Socrates. And the more we know, the less we know, right? The more we know that we don't know. If we were the attitude, we scientists, we're the attitude that, people should be educated, I know, well, then the people don't want to listen. Today is no longer the era where you have to go to the library to get a paper printed. Today, it's not even papers, it's fragments of information. And it takes nothing to be perceived as snobby.
Yeah.
And I'm afraid scientists need to adjust the way they communicate so that they not perceived as snobby. If we think that the administration and others are uneducated, because of course they're uneducated on oncology, they're uneducated on targeted therapies. I am uneducated on financial mechanisms. There's so many things I am not educated on, right? But the way we say it is very important. If we say, the public doesn't know, the public is uneducated, then the public doesn't want to listen. So I think part of it is getting to coming down from a lecture pedestal as a scientist and communicate science in a way that anybody can understand it so that people can understand why it's important.
Yeah.
Communicate the economic consequences of science, the financial consequences, and the consequence of society in a way that people understand it will make people accept it. I grew up in Italy. Until my parents were young, Mass, Italy's Christian, of course, the Pope is there, right? I grew up 2 hours south of Rome. Mass was being told in Latin. People couldn't understand it. There was a big revolution when Mass finally they decided that the mass would be told in the local language. People had to understand it. It feels like today we may be talking in Latin, and people don't understand what scientists are saying, and they say, "No, I don't want science. I want a chicken, fried chicken. I like fried chicken, but fried chicken is unhealthy for you because it causes this, and that." I don't understand what you're saying. I'm gonna get the fried chicken. If we don't explain to people who didn't study this subject why fried stuff you shouldn't eat too much, how can we get to an agreement? I think the same is on a larger scale for NIH, academic funding, biotech research, science, and so on. Part of it is a matter of communication coming down from the science
pedestal to the level. Excellent, brilliant. This has been a really fascinating conversation. I think that we all have a lot to think about and reflect on, a lot to be proud about, but also I think there's a lot to be done.
Yes.
And thank you so much, Giovanni, for coming and spending time with us and talking about the biotech world and what you do in it and how basic drug development takes a long time and is very complex, but at the same time, so important to society on so many dimensions. Thank you. My guest has been Dr. Giovanni Abbadessa from ModeX Therapeutics. I'm Ravi Sarathi. I hope you enjoyed this episode of International Business Today. Please subscribe and send us your comments and like our show and watch some of our other shows. And I hope to see you again in the future. Thank you very much.